think that, what we could come up with as a group was like, who would,
yeah.
I'll get that, I don't know. I was thinking out, but I don't know.
Somebody's graduated from lunch.
They are now, and they were, I think this, there were a few things that really excited us.
Yeah. All right.
They just want to be part of it. They just want to, they don't want to be treated as not eventful. Well, because they want to actually, they don't want us.
Yeah.
They want to be
So we'll see you again.
also not overestimating. All of you. I know he might be. Just be careful.
Hello, everyone. It's really a pleasure to have Colleen visit us. So Colleen and I kind of crossed paths in the same lab many moons ago. I'll tell you a little bit about her officially. She's an associate professor both in the Department of Psychiatry and the Neurosciences at the Medical University of South Carolina. And she leads a human addiction research laboratory dedicated to mapping, modulating frontal striatal systems that contribute to the cycle of initiation, use, and relapse among multiple alcohol substance abuse disorders. Colleen has done work in cocaine-dependent and smokers, certainly in alcohol users. Back in 2011, she got NIDA's Early Career Investigator Award, and since then has been kind of a shooting up. She has 4 NIH grants currently, all of them really combining that neuroimaging and brain stimulation to understand circuitry, but more importantly, modulating it in the right direction, which is a concept very dear to many of us, clinically as well as in the basic sciences. Her work has been highlighted even in popular press, National Geographic, Science Magazine, and Eureka Alerts. Colleen is also a very vested teacher and mentor, and a lot of outreach work. Some of her bright postdocs are actually U of M now. So we have a little bit of that science coming our way and that brilliance, I think. She just told us yesterday that she's actually accepted a position in Wake Forest, which is a huge next step, and I'm sure it's going to be a very successful one. On a personal note, the only thing that I would like to highlight is that other than being utterly impressed, having been knowing Colleen now for a little bit less than a decade, with all her work, is the incredible enthusiasm and positive energy that she generates is just unbelievable. And you can see her smiling, and she's I bet you're if you haven't interacted with her, you'll see it demonstrated live. This is kind of a unique aspects. Please.
Thank you, Ziad, very much for that introduction. And thank you all. Thank you for the invitation to come speak. I had never been to Minnesota 6 months ago, and I've now been here 3 times. And the weather has been gorgeous every time. Is it? But honestly, actually, in some ways, I feel like it's like coming home a little. I actually feel like I know a lot of you, and that I think speaks really well to your university, because it shows that not only are you all really collaborative with one another, but you have a really positive presence around the globe, which is awesome. So it really is sincerely a pleasure to be here. Thank you. What I'm going to talk to you a little bit about today is our work that we're using a technique called transcranial magnetic stimulation as a tool to decrease craving or cue reactivity. So cue reactivity is, I would argue, one of the most fundamental aspects of the human experience. And that's shown by this is sort of a mannerist painting of the Temptation of Adam done by Tintoretto, and this story is probably familiar to most of us in the room. You have Adam and Eve in the Garden of Eden, and Adam is being shown this shiny apple, and this apple is shiny and bright, and he's forced with this temptation and this decision. Should he take a bite of this beautifully appetizing, bright red apple and have pleasure right now? Or should he not? And should he withhold his response for a lifetime in happiness? Right? And so we all know how that story plays out. And I would say that that's part of our modern construct as well. So many of you, I may be the only one in the room that is faced with this, but I occasionally may be driving with my phone in the near vicinity near me, and I may occasionally see that somebody has texted me or an email has come through, and I'm forced with this temptation of, oh, should I look at it? And sometimes I don't know why I want to look at it, but should I look at it, or should I withhold that response, right? Because I know that that's actually what's better for me. That's true here anywhere. I was actually just in New York City, and I passed by this Dunkin' Donuts, which shows Dunkin' Donuts and this bright sign. And then right below it, it says the calorie range for 6 Dunkin' Donuts. And so right there in your face, you have this choice. And everybody makes a choice, and it's still doing great business. So there you go. And then, of course, also many of us probably are familiar with the experience. Maybe this will happen to you this week or this weekend, where you'll be out with friends and you'll be socializing, and maybe somebody in the group will say, "Oh, come on, one more round." And you think to yourself, "Ooh, should I have another drink, or should I go home?" And because going home is the wiser decision, I'll have to drive home. So this idea of cue reactivity is fundamental. It's not just related to our patients with substance use disorder. And what we could argue is that it's part of the balance between this appetitive drive and executive control, or ability to withhold a response. We've done a tremendous amount of clinical and preclinical imaging and sort of basic kind of fundamental electrophysiology over the last 20 years. And we've learned a lot about how the way the brain works and how these systems are organized in the brain. There's many, many models to discuss the difference between limbic circuitry and executive control circuitry. One common one is the difference between the mesolimbic circuitry, which is kind of shown here in red. This is circuitry that's mainly medial and ventral in the brain, and mediates craving, vigilance. Sometimes this is referred to as hot cognition. There's also the mesocortical circuitry. This often in human neuroimaging, we think about the dorsolateral prefrontal cortex a lot, but there are actually many sort of dorsal and tend to be lateral structures in the brain that are involved in this sort of top-down control. And then finally, there's the nigrostriatal system. These all have dopamine embedded in them in some way or another, involved in motor learning and control. I actually think this last system here, the purple one, which nigrostriatal, which I won't talk about much, is probably one of the most overlooked circuits in addiction. And I hope that the field starts to explore it more, because at the end of the day, in order to be a substance abuser, you need to pick up the cigarette and put it in your mouth, or pick up the alcohol and put it in your mouth. And so the motor system is So So if we look at this in a kind of simplified way, Alexander DeLong and Strick wrote a fairly influential paper in 1986 describing these frontal striatal thalamic loops. The title of that paper is Independent and Parallel Circuitry. And now in modern neuroscience, through a lot of really pioneering track tracing work, some of which Dr. Heilbronner has contributed to, we know that there's actually structural connections between these prefrontal, striatal, and thalamic circuits, and they're somatotopically preserved throughout the circuitry. So that's all really great, and if we think about those circuitries and we think about this competitive process between your appetitive drive and your executive control in terms of addiction, you may think that what causes some people to be a little more vulnerable to choosing to look at their cell phone or choosing to have that extra drink or going into Dunkin' Donuts is at that moment the ratio of connectivity or engagement of the executive control circuit is less than the engagement of the limbic arousal circuit. And so maybe what makes some people able to be social drinkers for all of their life and never have a problem with alcohol use disorder, but other people can't do that, is that there's a mismatch in the integrity of these two circuits. So this is kind of a hypothesis, which is not my own. It's science. We're all kind of standing on one another, and it's sort of a popularized idea. And if we think about that from a neural circuit therapeutic perspective, we have at least two strategies that we could try to do. We could try to increase the executive control circuit with some form of brain stimulation, or we could try to decrease this cue reactivity or limbic arousal circuit. And so for the rest of this talk, I'm going to kind of go through three little things. First, I'm going to teach you a little bit about what TMS is. I know actually Minnesota has an incredibly great concentration of neuromodulators. So in fact, many of you in the audience, this will be sort of brief review of stuff you already know. And then I'm going to tell you a little bit about how we're kind of using this technique to approach TMS development for substance use disorder. And then I'm going to show you some data hot off the press. You're the first audience seeing this data from the recent clinical trial we finished with TMS for alcohol use disorder. Okay, so what is TMS? Recently, TMS for addiction has gotten a lot of press. It's been in the cover of the National Geographic, and it was in Science Magazine. Some really influential leaders in the field have published really high-profile papers on it, and that can be very good. But I will also caution that a lot of the highest-profile papers of TMS recently, especially for cocaine, haven't been sham-controlled. And so somebody who's in this field, and I'm really excited about it, but I'm also a scientist. I want to be very careful that we all sort of temper our enthusiasm a little bit, and we make sure we do really rigorous science. So I'm going to show you how I've been thinking through the process as we go along. And hopefully this will just be a model for you all in your own thoughts. So it started with Michael Faraday. Many of you may remember Faraday from high school physics class. Faraday is one of my favorite scientists. He only had an elementary school education. He wasn't formally educated like many of the other sort of thought leaders of the time. And then potentially, interestingly, he gave Christmas lectures at a local university to children around Christmas, which I think is an endearing idea until you look up the titles of these lectures. So I envision this must have been like the kids of the professors that worked there. And this must be the most boring lecture to ever listen to, the chemical history of a candle. But alas, that was entertainment back in the 1800s. But what he showed that influenced our field the most is the law of inductance. And kind of to boil this down to its essence, you may remember the right-hand rule. And it's this idea that electricity and magnetism kind of hold hands. So if you have electricity and you run electricity through a wire, and then you coil that wire up, you'll induce a magnetic field that's perpendicular to the windings of the coil. So that's best known as the right-hand rule, right? So if you have the wires going this way, and then you coil them around, you're going to get a magnetic field that's perpendicular, right? And if you have this magnetic field and you turn it on and off, or up and down, positive and negative, you'll then induce an electrical field, which is perpendicular to the oscillating magnetic field. And it's that simple premise that is what the whole field of TMS is based upon. So to just kind of demonstrate quickly if these videos work, raise your hand if you've ever had TMS. Oh, see, I told you, you're a very educated audience. Excellent. Okay, awesome. So, so some of you again know this, but to those of you that don't, so here's a picture of, this is actually a high school student in my lab, and that's my arm there. And this is a TMS coil. I don't know if this will walk with me, but this is like a TMS coil with a big thick block of wood in the middle, and then another coil on the top. And this is because it's a sham-controlled coil that we'll use sometimes. And so what Rachel is going to do in a second is she's going to take this coil and she's going to bring it down closer and closer to my arm. And as she brings it closer to my arm, you'll see my hand begin to contract like this. And this is because your muscles are electrically sensitive. And you'll notice that before the coil is even touching my skin, my arm will begin to contract. So let's see. Hopefully it'll work. She's bringing it closer, and then you can see my hand is moving, and the coil's not even touching my arm. So that's an oops. We might go in an endless loop on that video. Sorry. So the next version of that is when you place the coil over the head, and so in this case. Okay, that's going to be annoying. So the next version, if you just trust me, is that when you take the coil and you place it over your head, so as you learned somewhere in your training, of course, that the whole body is somatotopically represented on your somatomotor cortex. And so if you place the TMS coil over sort of the hand area of your motor cortex, you can cause a contraction in your contralateral limb. Fairly reliable. If you move it up closer to your leg area, for example, you'll get contralateral movement in your leg. So it's very cool, and one of the reasons I like TMS more than many of the other non-invasive technologies is it happens every time. So as a scientist, our experiments fail way more than they work. We get results that are way more confusing all the time. But at the end of the day, you put a TMS coil on the motor cortex and the hand moves. So that's good. We're doing something. But we don't really honestly understand a whole lot more than that. A few things we understand. One, the principle of stimulation depth. So Maxwell wrote these equations which showed how a magnetic field will decay very quickly with distance. And so distance becomes really important in TMS treatment development. General rule of thumb is that the TMS pulse will affect about 2 centimeters deep. And that depends on a lot of sort of subtleties of the coil design. But it falls off very quickly. And that's going to be important for our substance users because a lot of them have cortical atrophy, which means their brain is much farther from their scalp. The second thing to remember is this principle of monosynaptic activity. So in this case, TMS, as you imagine, by putting the TMS coil on the motor cortex, you cause the arm to contract. But what that teaches us is actually that not only are you activating the neurons directly underneath the coil, but there's at least one other synapse in the spinal cord that the TMS induced pulse is able to transcend, right? So we know we can, by this general principle of collecting a motor evoked potential, we know we can affect 2 synapses down the line. So that's going to become very important when we think back about those prefrontal, striatal, thalamic circuits, because that's what we rely on to remember to sort of form hypotheses about modulating the basal ganglia. And you can show this in varying ways, using neuroimaging and using PET imaging. And so many sort of the pioneers of the field have demonstrated this really nicely. The dorsolateral prefrontal cortex tends to be the TMS treatment target for depression. TMS is FDA approved as a treatment for depression. Recently, a novel form of TMS called theta burst stimulation also received FDA approval as a treatment for depression. And all those studies are targeting the dorsolateral prefrontal cortex. I would argue that for addiction, the majority of the data from the preclinical side might suggest that we should target the medial prefrontal cortex. And this is because we know from electrophysiology and from optogenetics that if we can change activity in the medial prefrontal, specifically the infralimbic and prelimbic cortex, we can actually change drug self-administration. So why then, as clinical researchers, should we not try to manipulate activity in the medial prefrontal cortex to try to change drug use behavior in our users? The third sort of principle to remember is one of frequency dependence. So higher frequency stimulation generally tends to cause an LTP-like effect. So generally, high frequency stimulation like 10 hertz or 20 hertz increases cortical excitability, whereas low frequency stimulation like 1 hertz tends to decrease cortical excitability. Now I say that kind of with my fingers crossed because I think that we as a field are learning that it's not that simple. And so as the field grows more and more, I hope to never present this slide again because it's probably not true. But right now, this is what we know. So just remember that. The fourth principle is one of structural integrity. And so what we've started to learn is that, as I mentioned, TMS can affect 2 neurons, right? But in order for the signal to propagate from the first neurons or the first population of cells to the second population, you need to have an intact connection. And so the structural connectivity of the circuits that you're modulating is very important. And we have shown that in cocaine users, and then other groups have gone on to replicate that as well. So that's kind of an emerging principle also. So if we think about what we might do then, putting all that together, we could either try to increase activity in the executive control loop with something like 10 hertz or 20 hertz TMS to the DLPFC, or we could decrease activity in this limbic arousal or craving loop with something like 1 hertz TMS. There's also these techniques called theta burst, and so a technique called intermittent theta burst tends to cause excitation and behave like 10 hertz TMS, whereas continuous theta burst, which is what I'm going to talk about for the rest of the talk, tends to decrease activity. We recently, if you want to learn more about this, a group of us, a group of 77 authors, that's a hard feat in itself, agreed upon something, and we agreed upon something, and recently published a consensus paper on the state of the science of TMS and addiction. And so this is a really nice paper, and it kind of represents people from all over the world and some of the studies they're doing. It's worthwhile reading, if nothing else, for like the geopolitical landscape. So the Chinese tend to focus on certain things, and the Europeans tend to focus on certain things. It's very, very interesting. And there's varying sort of contributions. One of the things that holds the field back is that we're all using slightly different parameters, and we're looking at slightly different dependent measures. And this is true probably in every scientific field and discipline. And so you can see some people are looking at craving. That's the first column as their primary dependent measure. That's what we are using. But other really interesting aspects people are looking at are certainly consumption, which is what we're going to need to change in order to get FDA approval of TMS for addiction. Negative valence, positive valence, cognitive function. So our dependent measures are all over the place. But we're trying to get some consensus. Also, the field generally has been focusing on the DLPFC as the target for addiction. Again, kind of extending what's been done for depression. A lot of our substance users have comorbid depression or have high depression symptom severity. And so, in fact, maybe DLPFC is going to be the best target for some reason. I tend to like the MPFC, and I've just been working down that road, so that's what I'm going to tell you about. But I do kind of want to give sort of props to the people that are doing DLPFC work, because it is actually, as much as I don't want it to work, but it's working really pretty well. So Okay, so if you just want to go to sleep for the rest of the talk, the punchline here is that we've given a lot of TMS to the medial prefrontal cortex. So essentially, we're using the frontal pole as our target. It's kind of slightly over the left eyebrow in most people. At first, we thought my advisor, Mark George, is one of the leaders in this field, and he is willing to take risks, but he's always very cautious, and he's like, it's never gonna work. People aren't gonna be able to tolerate it. It's way more painful than you think. But in fact, I would say that we learned a lot of things about ramping the dose slowly in order to get people to acclimate to it. And now we've done, at MUSC, we've now done probably about 200 participants. Virginia Tech also has an active group. The Toronto group is doing it. So it's now really becoming a more feasible cortical target for TMS, which is great. I'm not going to show you all these steps, but just kind of outline how we've approached the field, potentially as a model for your own thoughts about developing non-invasive brain stimulation for a disease state. First, we had to figure out if we could reach the frontal pole in these cocaine users because of the cortical atrophy. Next, we had to figure out if we could differentially activate the medial prefrontal cortex to striatal circuit from the DLPFC cortex to striatal circuit. Because in fact, your head's I have a pretty small head, and so the coil is pretty big on my head. And so the distance between here and here is not very big. And with imaging, we don't have the CMRR, we just have me. So our imaging isn't sort of state of the art like you guys, and we smudge it a little and smooth it a little. So we didn't know if we would be able to differentially, activate those two circuits. Now we need to figure out which circuit to use, if we could engage it and move it just a little bit. And then finally, to run clinical trials for many sessions of TMS to see if we could actually change, drug use behavior. I'm going to kind of skip most of these steps and kind of try to get right to the clinical data, and talk a little bit about where we're going and where I think the field is headed. So one of our first things that we were challenged to do by our reviewer, the reviewers usually are pretty right, and a reviewer astutely wondered if we were actually even hitting the cortex with our TMS pulse. And so if you can see here, this is actually, I think I have a better slide in a second. So if you're not used to seeing the brain of a cocaine user, this is the brain of a cocaine user. So you can see there's a lot of atrophy around the brain. Those of you that are not brain imagers, all of this black space is hopefully not present in your brain. So here, I think probably about 40 years old. Their brain atrophy is more consistent. But what we learned is that even though there's a lot of focal enlargement, the distance between the apex of each cortical gyrus and the dura remain fairly stable. So they have gray matter loss, but in fact, the general skeleton remains fairly stable. So we generated an automated tool, which is freely available for anybody to use on the internet. And it will automatically calculate the distance between the scalp and the cortex, which we really hope the device manufacturers will start to integrate into their software, because it would be beautiful if TMS could begin to dose based upon the distance between the scalp and the cortex. Okay, so then we need to figure out a target. Like I said, I actually did some training in a preclinical lab, and so I'm always most of my scientific friends probably are preclinical scientists, and so I hang out with them, and they talk a lot about this literature. And so I was very biased to be interested in how, with optogenetically manipulating the medial prefrontal cortex, we can change drug self-administration behavior. And so we looked at a bunch of neuroimaging data that we had collected from my lab over time. We had 156 individuals, and they were exposed to blocks of drug cues that were tailored to their specific drug of use, and neutral cues and rest cues. And we compiled all the data together, and we said, okay, transdiagnostically, what brain areas are activated by the drug cues versus the neutral cues? And what we found is that reliably there was activity here in the medial prefrontal cortex, as well as laterally in sort of the anterior insula area. When we took that data and we divided it down into the drug of use, all three groups had loci that were present here in the medial prefrontal cortex. Everyone looks at this picture, I made a mistake. There's actually red underneath there. Everyone thinks that it's just the green. But all three groups are here. And then the cocaine users also tend to have, the cocaine users also tend to have activity in the insula anterior bilaterally, but the alcohol users and the smokers did not. When we take that data and we look at areas of maximum activity, we can then figure out what cortical area, if we apply a TMS pulse and we model the distribution of the pulse. I know Alexander Opitz is here at University of Minnesota, and he's one of the leaders in this. We didn't know him then. But we just basically did a back projection on the cortex, and we found that the left frontal pole would capture the maximal number of hotspots. And so that's where we determined that we would put our TMS coil. And it's going to depend upon the shape of the coil. So now there's more and more coil designs coming out, and that the coil design itself might shape where you want to target with your own clinical trial. Again, this is less about why we decided to do what we did, but maybe to kind of help you to think through it a little. One question I often get asked is, are you targeting the ventral medial prefrontal cortex or the orbital frontal cortex? And, I don't really know. Sarah knows the answer to this question. But we think, I've published both ways, unfortunately. I think by and large, we are just going to start saying that we're targeting the frontal pole, because that's probably just the most accurate thing that we're doing. But in fact, in the case of humans, the orbital frontal cortex and the ventral medial prefrontal cortex have a lot of overlap. And you can get into the details of, are you medial or are you lateral? And I would argue TMS is a fairly blunt tool. So that's just kind of, if you're interested in that. In contrast, the nomenclature continues to be a problem in the field. Jonathan Downer, who's a really influential psychiatrist at University of Toronto, has published some important papers, and he is also stimulating the medial prefrontal cortex. But in fact, he's stimulating the dorsal medial prefrontal cortex, which is actually fundamentally different than the ventral medial prefrontal cortex. These are really important things. And they're both different from the dorsolateral prefrontal cortex, which is the area that's mainly being stimulated for depression. And then there's a new kid on the block. TMS was just FDA approved for treating obsessive compulsive disorder. This is from a special type of coil. It's produced by one manufacturer, and it's called deep TMS, but it's a little bit of a misnomer. It really should just kind of be called, I would argue, like wide and deep TMS. So it takes kind of a whole chunk of the brain out, like a big ice cream scoop. So it's unclear exactly where in the brain it's stimulating, but it's probably a little bit of the dorsal medial prefrontal cortex and a little bit of the supplementary motor area and a little bit of the cingulate. So these are this is a summary of like the main targets that are being used for TMS in psychiatric disorders at the moment. Okay, so then the next step is, okay, we have a target. Now, can we change the brain? Well, randomized, double-blind randomized, sham-controlled trials are long and they're expensive. And as you can see here, there's a lot that we don't know. And so we have kind of followed the NIMH sort of lead or suggestion, and we've started doing target engagement studies where in a single day, we basically put someone in the scanner, and then we tap on their head with TMS, and then we put them right back in the scanner. And in this case, that's what we did. Individuals, maybe this will work. Individuals went into the scanner. They saw drug cues tailored to their drug of choice. Then they came out of the scanner. They received real or sham TMS, and then they went right back in the scanner. Each person came back twice. One time they got real, one time they got sham. We treated at 110% of resting motor threshold. We gave a bunch of TMS. And this was a type of TMS that we think is causing a decrease in cortical excitability, something called continuous theta burst stimulation. Another important thing I think that will be increasingly important as we move forward is that we showed everyone cues. So there's really good data in PTSD and also in smoking trials that if an individual is exposed to cues first, so their brain is primed, the effect of TMS is stronger than if they're not primed. And in fact, the recent OCD FDA approval requires that the individual is primed. So they take, if you're worried about things being dirty, they take your purse and they hold it slightly above the ground. It's very cruel, actually. But you have to do this priming to be compliant with the FDA rules. So priming is probably very important. As a neuroscientist, that makes perfect sense. So a primed circuit we know is more plastic than an unprimed circuit. So we always show them cues before we're delivering the TMS. This is the protocol. Looks like this. I'm not going to play it, but we've also started publishing the standard operating procedures with most of our clinical trials papers to help people try to replicate them. There's a lot of subtleties in TMS, like what does the room look like, what's the position and angle of the coil. And so we've also started kind of creating a database of videos that are online, and we have a YouTube page if you want to go see them, or I can always send them to you if you ever want them. Again, I hope that you kind of get what we're trying to do here is that we're trying to get as much information out as possible such that we can all try to replicate one another and extend the field, because the variance, I think, is what's holding us back. Okay, so what happens? The first time that we started, TMS, repetitive TMS had only been given to cocaine users in one previous study. So it was a little bit risky. Some people worried that we were going to cause a seizure. But in fact, cocaine users, it's harder if they're not actively high. It's actually harder to cause a seizure in them because their motor threshold is much higher. It takes actually a lot more energy to get their brains to fire than somebody who's not a cocaine user. But we started very slowly at first. We put individuals in the scanner. In this case, we used a technique called interleave TMS, fMRI, which I'm not talking about today, but essentially we take a TMS coil in the MRI environment. We provide single pulses of TMS and we can probe the neural circuitry. It's really like a beautiful tool to help you, allow you to kind of ask these really important questions, which is great. And then we gave them real or sham TMS, and then we tapped on their brain again to see how the circuit would change. And what we found is that real or sham TMS is a relatively small sample, but we still found significant decreases in TMS pulse-evoked connectivity between the medial prefrontal cortex, the nucleus accumbens, and the orbital frontal cortex. So that was kind of, that was good. At least it was a good start. But it's a really small sample size. You can't really make much of it. It was just kind of a proof of principle. So then we did a larger study. In this case, it was single-blind, sham-controlled. We had 49 individuals. 25 were cocaine users, and 24 were alcohol users. Same design. When we used single pulses of TMS, this interleave TMS technique, as our dependent measure, we found that real TMS decreased activity in the medial prefrontal cortex. Here you can see this is the ventral side of the brain, so this is the orbital frontal cortex underneath, as well as in the basal ganglia here. You can see that this is the putamen bilaterally, and then moving back. So that was real versus sham TMS in 25 individuals within subject controlled. So that was pretty exciting. These are the actual data here. And we found that the same thing was true in our alcohol users. These are the data here. So the last slide was cocaine users, and then it also generalized to alcohol users. Alcohol users, interestingly, didn't have as much striatal modulation as the cocaine users, but both had this very large ventral medial prefrontal cortex difference. That's great. That's still a little bit of like a basic science question. I work in a psychiatry department like you guys, and I'm surrounded by MDs, which is, for someone like me, a beautiful thing, actually, because it's a really nice relationship. Because the clinicians always kind of bring you home from all of your cool neuroimaging, tricks. And they say like, okay, great, but how is that going to do anything for my patients? And so in this case, we also looked at cue reactivity. So can we actually change the brain's response to cocaine cues or to alcohol cues? And in this case here, the way this is shown, this was done by outstanding postdoc in my lab, who's now an assistant professor at Columbia, Tanisha Kearney-Ramos. And you'll see here, first you'll see the baseline functional connectivity between the medial prefrontal cortex and the striatum. So here at baseline, this is a technique called PPI analysis. So people are looking at cues versus neutral cues versus drug cues. And at baseline, in the cocaine users, there's high functional connectivity between the medial prefrontal cortex and the left putamen, as well as the insula. In the alcohol users, there's really high connectivity also between the medial prefrontal cortex and the striatum, as well as the insula. Following TMS, in both groups, we have a specific decrease in functional connectivity between the MPFC and the left caudate and putamen, as well as the insula. So this kind of transdiagnostic nature was really very kind of heartwarming, or whatever the word is. Felt good to get those data. So that was nice, and she published those data. What we didn't find, expected, was no difference in craving. So in fact, when the individuals got sham TMS and when they got real TMS, the reality is that their self-reported craving didn't change. Now, I say that with the caveat that one reason may be because we just gave a single session of TMS. A single session of TMS certainly doesn't cure depression, and it certainly doesn't cure OCD. So why would we even expect that it's going to change something like cue reactivity in individuals that have been using drugs for 20 years? Alternatively, it may just be because our measures of craving are pretty terrible. Basically, we ask them not much more sophisticated than how much are you craving on a scale of 1 to 10. And for the most part, they look at you and essentially say, what's craving? Right? So craving is a very academic construct. And I think actually some people in this room are coming up with much better ways to assess what's going on with individuals. And I think some of like the neuroprism stuff, I think we need to get much better at our assessment tools in order to really kind of measure what we're changing here. So then finally, we started a series of clinical trials. So we showed target engagement. We showed that we could change the circuit. We picked a target, identified it. Target engagement showed that we could change it. And now we're going to a clinical trial, which is a much bigger effort. And in this case, we did a 10-day trial, real and sham controlled. I'm going to show you the data for alcohol. We've also done this for cocaine users, but we haven't finished yet. And basically, we have a really good intensive outpatient treatment program at MUSC, where individuals come in and they receive about 4 to 5 weeks of therapy, which they receive about 3 hours a day. We're giving them TMS pretty much before their intensive outpatient treatment programs for 10 days. And so what we did is we brought them in, and on their first week of being part of this intensive outpatient treatment program, we put them in the scanner and we showed them drug cues versus neutral cues. And then we randomized them to receive real or sham TMS while they were in the program. The program lasts about a month. We put them in the scanner again. We showed them cues. And then we did a 1-month and a 2-month follow-up, collecting neuroimaging data at all of those time points. So anybody that has ever done a neuroimaging study knows that that's really expensive, number one. So we scanned them at all of these time points. It was a total of 51 individuals were randomized, 520 total participant visits, 469 TMS sessions, because some people dropped out, and 160 MRI scans. So it's a lot of work, like I said, and it's only 50 people, which is the biggest study. This is the biggest study I've ever done thus far, and it's only 50 people. So the next step, if it turns out to work in the right way, is clearly to have multiple sites, because one site certainly can't make that much of an impact on the field. And if you're interested, these are alcohol users. This is kind of just generally what they looked like. The age is about 46 years old. I'm not sure, this is actually not the full cohort, but generally these things remain true. So the age is about 46 on average. They tend to be mostly men. Interestingly, we also had the VA as a site for this. I'm only showing you the data for MUSC. We also had the VA as a site. And the VA data looks so different than the MUSC data, that in fact we'll publish them together, but it's almost, they're like apples and oranges, which is its own interesting thing. But this is the first time I'm showing these data to anybody, so bear with me. So this is enrollment in the intensive outpatient treatment program. So their engagement in the treatment program itself. So at baseline, we have all individuals are enrolled. The blue line are the people that receive real TMS. The orange line are the people that receive sham TMS. So for the first month, they're receiving TMS plus some type plus CBT. So it's not exclusively cognitive behavioral therapy, but our therapists tend to sort of bend that way in their own treatment. But it really is kind of treatment as usual for these individuals. Nobody was on naltrexone, which is important because these are alcohol users. And then we follow them up at 1 month after, so we have a 1 month time point, and we still maintain pretty good enrollment in both groups. So I would argue this is just the effect essentially of our treatment program. So it keeps people enrolled, which is great. Then the next step is what happens 1 month later when they're supposed to be coming back for their continuity visits, and then 2 months later when they come back for their continuity visits. And you'll see the dropout rate tends to be a bit higher in the sham group. So the sham group doesn't stay as engaged in the treatment program as the group that received real. When you look at sobriety, you see this similar kind of pattern where at baseline, so some individuals, interestingly, were sober when they started treatment. You see that with alcohol users in particular, which is interesting. They've achieved sobriety, and then they go enroll in treatment. After the initial treatment phase, sobriety was relatively high. And you can see here, we may not have followed out far enough, but the group that receives sham tends to be relapsing at a higher rate than the groups that receiving real. So these are all, I consider this a victory. We had no serious adverse events. We had no unexpected adverse events. The biggest side effect was getting a headache, which we had anticipated. We had no seizures, we had no syncopal events, or no sort of unexpected things that people reported to us. So I think this is a really good start, and it's a positive signal. I'm happy to say that we received an 8 percentile on an RO1 to kind of do this in a more formal way. So hopefully we'll have some more definitive data moving forward. But we're moving in the right direction. So I think it's a really exciting time for the field. This is the neuroimaging data. This is kind of just a first pass at it. Again, so I had a graduate student do this for me last week, so I haven't really checked it carefully. But at baseline, the brain reactivity to alcohol cues is really high in the areas we expect, like the medial prefrontal cortex and the anterior cingulate. So that's what we expect, this like cue-evoked activity. After treatment, you still see in both groups elevated activity in the medial prefrontal cortex. So when we're showing them alcohol, their brains are still turning on. That said, if you remember the previous graph, they're achieving sobriety the best that they're going to achieve. So their brain is still turning on, even though they're not drinking. In our first follow-up visit of the individuals that came back, we seemed to lose some of the signal associated with cues in the group that was receiving real TMS, whereas some of the signal still lingered in the group receiving sham. And the same was true at the 2-month follow-up. So I'm not really ready to interpret these data too much because we kind of just put them together. But what I will say is it makes me feel good that at baseline we see what we've shown in our other studies and other people in the field have shown, is that we get this robust baseline activity to alcohol cues. And it looks like this activity may remain stable. It also looks like it might not track perfectly with sobriety. These are all really interesting questions. Okay, so our vision is that moving forward, we're going to be able to hopefully kind of leverage this large international group of people to kind of come up with a consensus and do some multi-site clinical trials. I really only talked about the medial prefrontal cortex as a target, but as I mentioned, the DLPFC is also really a pretty good target. So hopefully doing some comparisons and breaking them down between groups will be good. As I spoke to a few of you about in our meetings this morning, I think, and certainly any of you that are clinicians just inherently know this, addiction is not a monolith. And in fact, it's a process that evolves over time. In the earlier stages of addiction, you tend to have, there tends to be more of like a reward-based responding or limbic-based responding, even fear or withdrawal, anxiety. Whereas over time, it becomes more of a habitual responding. And we know in the brain, those are different neural circuits. And so if we're going to be smart and come up with some intelligent, comprehensive brain stimulation-based treatment intervention for these individuals, we should probably think about where they are in their stage of the addiction cycle. So that's an important thing to think about. We also have found, so Tanisha Kearney-Ramos again we recently published a study which shows that there's state-dependent effects. And I know Alec Widge, who's here, also feels very strongly about this, that in fact, the TMS signal tends to, in our hands, individuals that had high cue reactivity at baseline, CTBS tend to decrease their cue reactivity. That's what we wanted. Individuals that had low cue reactivity at baseline, CTBS tended to increase their cue reactivity, which was not what we wanted. So that's interesting. And that opens up a really nice box of questions regarding individual variability and the state dependence effects of TMS. And then another thing, in depression literature recently, there's been a lot of emphasis on biotypes. So if any of you know or are familiar with this paper, it's been talked a lot about recently, and that among depression individuals, there are potentially these biotypes that exist, right? So there are essentially phenotypes. It's a really complicated paper to read. And so the Cliff Notes version, in my opinion, is that in fact, there's different sort of subtypes of depression, which map onto symptoms and brain networks. And in fact, TMS differentially affects them. And so we could potentially use brain imaging and this behavioral-based phenotyping to guide our TMS target selection. So if that's true in depression, it should definitely, there's no reason it wouldn't be true in addiction, of course. And so that's where I think that a lot of us in the field are moving, is looking at sort of perhaps impulsivity versus compulsive aspects of drug use behavior and where we might differentially choose a target for TMS. So again, the next step, what will be the, what site will be best? Does it depend upon the stage of the addiction, or does it depend on the symptom severity? So that's just to kind of in closing, thanks for paying attention. I just want to kind of point out, hopefully I've educated you a little on how we have thought through this very complicated problem to inform your own decision making. But we know so little. The questions that we have to answer is, where is the best location? What's the best number of pulses? How many days do you have to give TMS? So there's a big emphasis in the field now of depression to squeeze a lot of sessions into one day. And that might be great. It might be particularly good for people that are maybe suicidal or need an acute intervention. But there's also some emerging data to suggest that when you squeeze it, the durability is less. So all of these really important questions we don't know the answer to. As a scientist, it's great. There's endless grant writing opportunities. So anyway, I just wanted to kind of use that as a recruiting tool for those of you that are interested in going into neuromodulation. It's a really great area, and addiction is a sort of a growing one. And that's all. These are the people that have contributed to all of our work. Two of my graduate students, as Dr. Nahas mentioned, are here as postdocs. I don't know if either of them are here, but if you see them, they're both really smart. Say hello. Logan Dowdle is at CMRR. He loves pre-processing like no one I've ever seen. And Daniel Lynch is working with Bernadette Gillick doing neuromodulation in pediatric stroke patients. And then just one final thing is Ziad mentioned, starting this November, I'll be moving to Wake Forest University, where I have the opportunity to direct a new center on electromagnetic therapeutics. So we'll be doing our addiction brain stimulation work there, a lot of the things that I just talked to you about. It's actually a partnership with the Comprehensive Cancer Center. There's some really interesting data to suggest that low-frequency pulsed magnetic fields can slow tumor growth. So I'm excited about that new thing, too. Anyway, if you have any questions, feel free to contact me. Thanks for your attention.
So I was right. It was wonderful. It's just a small anecdote. So 20 years ago, I think I have the reason why Mark George said it would hurt. We were two fellows in his lab, and my counterpart wanted to do studies in anxiety disorders, and he thought he wanted to target orbital frontal. And the problem was that he was more anxious than his patient, and he never walked them through the protocol, and that lingered in this background, both he and I. It's really intolerable. Yeah. But anyway, that's 20 years ago. Questions?
Beautiful talk and such interesting and important work. So I was wondering if you could compare and contrast what you've done with the Banshee Lab studies on cocaine users.
Yeah, sure. So for those of you that don't know, so Antonello Banshee was the head of the NIDA intramural research program. He's a very influential preclinical scientist, and actually is a neurologist by training, and has recruited basically created a storm of TMS research in Italy. And the Italians have this really great program, actually. So one big contrast is that in Italy, almost all of the cocaine users that they're studying are using powdered cocaine. They tend to be really wealthy. They fly in from places like Bali to have their TMS done, which is a little different than in Charleston, where we get them from low-income housing. So there's a little bit of socioeconomic difference. But they have really done some extremely influential work where they're targeting the dorsolateral prefrontal cortex with high-frequency TMS. It's very similar to the depression protocol. And they've shown an incredible, so that there's no sham TMS intervention in their studies. So that's a very large caveat. But I will say they're treating with medication as usual in their other group. And so when they have medication as usual in one group and real TMS in the other group, the group that's receiving real TMS has unbelievable abstinence rates. So yes, that's
unbelievable.
No, that's a really
very
poor
high
choice of
abstinence
words.
rates. Is that right?
I definitely take that off the record. Very believable, but incredibly impressive. So yeah, what were you going to say?
Is it TMS plus treatment as usual, or TMS alone versus treatment as usual?
TMS plus treatment as usual versus treatment as usual.
To your knowledge, are they measuring Cue reactivity or anything? That's not a feature of their studies so far.
In the initial studies that were published, so Terranio et al. Is the paper. It was like European Journal of Neuroscience. It's what led to a big National Geographic article. They didn't have any brain imaging or Cue reactivity, but in fact now Italian Giovanni Marinati, he is actually leading a really good study. It began last year where they have real versus sham TMS. They're doing a whole suite of neuroimaging things at baseline and at the follow-up portion. I think they're doing maybe 20 sessions of TMS. So I think we will get more rigorous data moving forward. Still the dorsolateral prefrontal cortex, high frequency.
So if this form of TMS that you're using is more uncomfortable, how do you know that the sham is a real sham? I mean, can cannot the patients tell that they're getting the real painful TMS versus the sham?
Yep, great question. So I didn't show that too much in the slides, but I can show you if you want. We're using a device which the device manufacturer has paid a lot of attention to creating a sham embedded in it. And so we have electrodes that go underneath the forehead, go on the forehead, and the TMS coil goes on top of the electrodes. And when the TMS coil, there's a gyroscope built into the TMS coil. And so this also explains the blinding. So the research assistant just puts in this little USB drive, and the USB drive has a randomization code which says real or sham. And when when the real side is down, the electrodes are off. And when the real side is up, like facing away from the patient, then the electrodes are on. And the electrodes cause a type of stimulation which, if you're not an experienced TMS person, you can't tell the difference. So we ask them to rate after each week, I think, we have them tell us if they think they receive real or sham, how confident they are, and we have them write a sentence about why they think it is. So the short story is that they can't tell the difference. And generally, they think everything is real because it's kind of painful. They say things like, well, but it's got to work because I feel it, you know.
Thank you for a beautiful talk. So my question is, in your initial target engagement studies, I'm curious to just hear more about the rationale for going right to theta burst versus doing 1 hertz studies.
Yeah. I think 1 hertz should be done, actually. That's a short answer. My rationale at the time was that theta burst was kind of new. Now we know that theta burst is more variable than we thought before. And so at the time, it seemed like it was new. So it was based on sort of theta rhythm in the brain as an endogenous neural rhythm. It's associated with learning and memory. Again, for me as a neuroscientist, it made sense. It was patterned. It's more efficient. So that's why we went with it. It also is super potent. So it only takes about 40 seconds to get the same effect on motor cortex as it would for about 12 minutes of 1 hertz. So it's really fast. So that's kind of why we did it. That said, if I had I would definitely think a 1 hertz study would be great.
Yeah, great talk. I'm just echoing what everyone's been saying. You did such a great job of being able to take the uninitiated kind of through the principles, as well as making it super interesting for people who've been thinking about the area. I'm just curious, I mean, it's going to be one of two things, right? One of two kind of forks in the road coming up. Either there's going to be biotypes, and there's suggestive evidence that there are biotypes in the different substance use disorders. And there's going to be, biotypes that are defined by pre-existing neural system functional configurations, which will guide then what are the appropriate targets and how one should be thinking about engaging those targets through TMS. And or there's going to be people where it's not going to be one target or another, right? I mean, just based on your talk, one could conceive of layering. TMS that is looking at or seeking to modulate different targets in different ways. And I was wondering what your thinking was about that latter strategy.
Yes, I think that's a great point. And I think I'm probably not in the room that has the most expertise with this, but I'm sure that the depression providers already often will, sometimes they'll stimulate the left and then they'll give a little bit of the right. Maybe people are doing all kinds of combinations. Like I said, so there are definitely people in the audience nodding that know way more than me. And so in as much as that's that's the way that it's playing out in depression, I expect that would be true in addiction. I think yes, I would actually lean on the sort of addiction psychiatrist to answer that question, honestly.
Have there been any studies, even pilot studies, which attempt to sort of first focus on essentially more medial parts of frontal cortex and to reduce cue reactivity, and then to focus, say, subsequently on dorsolateral?
To my knowledge, nobody's done that yet. That's a great idea. I think we're still at the stage where we're like, ooh, does it do anything? But hopefully we're getting to the point now where there's enough data from at least these 2 sites that people can begin to think about that.
We must remember that today is 9/11, a disaster of 18 years ago. And per your first observation of the ratio between ego control and impulse control, we wonder about that balance in those people who were flying those airplanes. A question. But back to this study. In traditional treatment for these disorders, the whole aspect of aftercare is a major phenomenon in whether people make it or don't.
Yep.
In terms of sobriety. In the 2 studies that you did, not the studies, the 1 month and 2 months follow-up, can you speak to what else happened to these people who were, I gather, finished with the treatment program and then went out about their lives, and what happened in terms of other influences? Like the common thing, of course, is 12-step programs for treatment, so on.
Yeah, that's a really great question. We haven't gotten sort of, so that if I to paraphrase your question a little, so the question is after this intensive outpatient treatment program where we follow them up 1 month and 2 months later, what happens to them in that 1 month? So all we know, not all I know right now at this moment is that they came back for their MUSC academic-based continuity visits. And so those are, so the way that it works, I think, insurance, our program is paid by, accepts insurance, and insurance accepts a certain number of times. They have to come back, they come back at least twice a month for continuity visits through the MUSC program. But we do have some people that opt to also do AA or NA during that time. So, Alcoholics Anonymous. And I haven't gotten granular deep down in the data to see who did that and who didn't do that, but yes.
So I wanted to ask you about the imaging data that you presented with activation of the insula. At least in depression, I feel like insula is way underrated. And it is a target, potentially a target also in terms of monitoring homeostasis, et cetera, et cetera. Can you comment on that difference, or maybe speculate about the differences between the cocaine and the alcohol when it comes to the insula? And do you see that as being a potential, is it transsynaptically being modulated? Let's not make it as a direct target, but.
Yeah. I don't know. So I think the insula is not underrated in addiction. So I do think a lot of people talk about the insula and addiction right now, and there was this study maybe like 10 years ago where people that happen to have a stroke and they enter insula spontaneously stop smoking. And so now Michael Fox has built his career around taking people that have had strokes and looked at various things. And I think that, and I do think the insula is important. I do also know that it doesn't show up in alcohol literature as much as it shows up in cocaine literature. And it shows up in smoking literature, but I can't say confidently that's not just because it's a bias in what we're asking. Cocaine people are primed to ask about the insula. And I think, yeah. I know that's a really cop-out answer, but.
Are you modulating it?
Oh, that with the MPFC? Yeah. Probably not directly. But there's an H coil design that can reach the insula. So that's one idea. And actually the Columbia group, Deanna Martinez, has done some studies with this H coil modulating the insula in cocaine users. That was published last year. And they actually found really good data on cocaine self-administration in an inpatient setting with this coil, which really, hits the anterior insula, but actually also hits all the brain all the way down. So it's either the insula or it's the inferior frontal gyrus.
One of the things the DBS world has sort of converged around is the idea that very fine differences in placement, in electrode placement and individual anatomy can have a huge impact on outcomes. And I wonder, in your opinion, is TMS just way too broad and it's just too big, too much, too non-specific to even is individual targeting via like structural or functional connectivity worth it? Or is it just like a paddle and then you're just done? It's like close enough above the eyebrow.
So at some point I'll send you this paper I wrote. Actually, I went to ACNP and got in an awesome discussion about this with a bunch of colleagues. Is TMS blunt or precise? And so right, if you so we can do such elegant things with tools that can go down and hit like small populations of neurons, right? And we do that in humans with DBS. And the other end of the spectrum is we give ECT. And bilateral ECT, correct me if I'm wrong, tends to do pretty darn well. So we have this spectrum where broad seems to work really well sometimes, and really fine seems to work really well sometimes, and TMS is somewhere in the middle. So the people that make these big coils, these H-type coils, they use that as their explanation all the time. They say we don't have to be precise. In fact, we just get a bunch of brain and hope that we're right. Which, it's not elegant, but maybe it'll work. So as a neuroscientist, it makes me cringe a little bit. But I think it's a worthy question. How much do we need to use neuronavigation or online neuronavigation, given the precision of the tool? It's an open question.
Clinically, it hasn't made a difference.
Oh, really? Right. Whether you're precise or you're blunt.
The variability that you started off, your thought,
we have so much unknown about these variabilities that we still are not very specific in our targeting.
Oh, right. Yes.
And the advantage of larger coil is essentially saying, I really don't know where my spot is. I have a better chances of going at it.
Yes.
But even then, clinically, we're not finding a difference. We were talking about it earlier, between the H-coil wide approach TMS and the figure of 8 tip of your thumb kind of effect. We're not finding clinical differences in a depressed population, in part because of the non-specificity of who we're including in treatment.
Oh, yeah. That's great. I think the field, so certainly like everybody in their grants these days does neuronavigation to be really precise with the targeting, get you good innovation points, But in fact, I would argue that in some ways, what if you're wrong? That means you're wrong on exactly that same spot for 30 sessions. If you don't measure it, you're going to kind of get the field around it. And so maybe actually you should hedge your bets a little and not worry about the precision.
The outcome in the new data you showed us?
We don't know yet. Yeah.
Do we know anything about those fine distinctions in, for instance, the effect of naltrexone on cue reactivity? Does naltrexone modulate cue reactivity according to whether or not someone's a high or low baseline cue reactor, as it were?
I don't know the answer to that. But so Ray Anton and Joe Schacht did a study where they did neuroimaging of a big study, the combined study, where they'd used naltrexone plus behavioral interventions, I believe. And they collected neuroimaging data. I'm not sure if they looked at that or not, but yeah, that's a great question.
You want to plug your talk tomorrow?
Pardon? Oh, yeah. If you're around and there's a big addiction symposium tomorrow, which hopefully you all know about. It should be great. They're leading it, I think. And I will be presenting again, but I won't be presenting these data. We actually have really interesting new data, on TMS as a tool for pain, potentially as a way to decrease the likelihood that people use opiates. So I'll be spending a little time talking about that if you're interested. Thanks.
Thank you very much.
I knew Leighton McCurdy.
Oh, really?
Do you know what's come of him?
Yes, he still comes to our division meetings.
Oh, does he?
occasionally.
He's still
Yes.
active and at it, huh?
He sure is, and he's smart as well.
And David Klein, you might you can remember,
Okay.
say hello to from
I
way
will.
back in the 70s.
Okay. Oh, yes.
They were academic psychiatrists too.
Oh, great. Well, I will try to remember.
You have been disconnected from the meeting.